Hmm. Good. So A contains the most of catalase enzyme, B has catalase enzyme but lesser than A, and plant C has no, tissue C has no catalase enzyme. Good. Suggest why the disc paper were left on the cut surface on the plant tissue for the same time of length. So first of all, we need to place the, okay. Okay, but why the time for a specific time, like same time, time to have comparable result. Okay, and if it's of 2 mark, then what should else be written? Okay, okay. Your concept is right, but we have to rephrase it. It's so that it can absorb same volume of liquid. Okay, so absorption is correct, but what it needs to be absorbed, it need to absorb the same volume of liquid and to have a comparable results. Okay, next one is what is a suitable control. Now you are using something. So okay, wait, no. Contr- Control mean, okay, we have already discussed this, that control group means that whatever you are investigating shall be absent. So you can use water instead of, yes. So we can use a denatured one, boiled tissues. We can use boiled tissues like to denatured one. And second thing, we can use water instead of hydrogen peroxide. Wait. No. Okay, that whatever you're talking about is like controlled variable. I'm not talking about variable. I've told you earlier as well that there are two groups when we carry out experiment. One is called experimental group in which we have to change only one variable at a time. Okay? While the control group is the one which is not provided with the independent variable. Okay? So independent variables should be absent. So it can be boiled tissue instead of these tissues. We can just boil them so that to represent that the denatured enzymes. Okay. Or the second thing is we can even use water in the disc. So so first of all we put the disc on the tissue so that the enzymes can be absorbed. So instead of enzymes we can use water, just water. So no enzyme. Okay. A length is given for a certain plant, so you have to calculate the length from D to E, that you can easily do. So your scale gives you a value of centimeter, you have to multiply it with 10. 10, yes, to have a sorry, length in millimeters. Then divide it with magnification to have a actual length. Clear? Okay, the population of this plant grows by each plant divided into smaller plants. These smaller plants then grow new leaves and divided again. Okay. Some students decided to investigate the growth of lamina plant. They place 6 plants in the small beaker containing nutrients in distilled water. Now, why we have used distilled water? Why we're not using tap water? Right. So we because we are using the distilled nutrients, now we can add the nutrients as per our choice. So why the nutrients are added? No, why the nutrients are added? So nutrients should be added for growth. Okay. The next one is how many plants, how many seeds can you see over here? So you have to count them. Okay. Let's say let's say one with three. I'm counting the one which have 3 leaves. One, two, three, four, five, six, 7, 8, right? So 8 into 3. No, we have to calculate all. I'm just giving you like a shorter way. Now what you can do, calculate for 4. 1, 2, 3, and 4. 1, 2, 3, and 4. So 4 have 4, so it is 24 plus 2 plus 16. So what would be your value? I guess it's 42. So there are total 42 leaves which you need to write here. Okay, it would be, let me write here 42. Okay, now you have to make a graph and it's a line graph. So whenever you make a line graph, keep in mind the like you have to write the dependent variable and independent variable. So let's say time should be taken on x-axis and this one is y-axis. So labeling should be same, time slash days and total number of leaves. Okay, and then what you need to do, you need to make 7 points according to these 7 points: 0, 2, 3, 4, 5, 6, 7. There is no one, keep this thing in mind, there is no one. Okay, there is no one in the day, like value for day one is not given. Oh my god, my battery is going to be dead soon. Just a minute. Why? 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 Wait, I couldn't put the battery. What's the issue? Hmm? Oh my god. Oh, many people ask me the same thing. It's almost two years back. It's not new. Okay. Okay, the other thing that I was explaining, okay, x-axis, y-axis, and then we have to put dot with a circle, or rather I would suggest put a cross. Okay, and the said the coinciding point or the center of cross should be combining should be joined with the cross of other, sorry, coinciding point of other cross, and it should be a ruled line, means you have to use a scale. Okay, and then you have to calculate the number of leaves for day one, which is not given. So simply place a line on y-axis and x-axis wherever there is coinciding point, that is your day one. And when they ask you that you have to show your work, then you have to show these lines on x-axis and y-axis which you place to find the value. Okay, next one is predict the shape of the graph after day 7. So what do you think, would it be still increasing or decreasing or constant? So what do you think? Like if it's straight, if it's a straight line, let's say if this line is like this, so what should be after day 7? What do you think? Would it be straight or still increasing? No, it won't decrease, no. Okay. It can become constant. Okay, now why it's becoming constant, the prediction is, let's say it won't it would be level off. Okay, mean it would be straight line, it would be level off. Why it is becoming constant? Because maybe the nutrients are used up. This can be one explanation, right? The nutrients can, sorry, no, we are checking the effect of nutrients, right? So nutrients are have a direct impact on growth. So if there are no nutrients, there is no growth. Okay. No, because as no such information is given, so we cannot write about the color of the leaf. Okay, we need to stick whatever they have given us. Okay, next, suggest one other method that the student could use to measure the growth of lamina. Okay, the one thing that they have used is counting the leaves. What other method can be used? They have used counting, they have used the like counting, like let's count the number of leaves. What can we do, like if we want to change the method? No, we don't, we are not using the same method, we have to use some other method. Hmm. Exactly, we can measure the mass, we can measure the surface area. Okay, but not the number of leaves. They have already used this method, we have to give some other method. Okay, the next point is, now we have to plan the investigation of different concentration of nutrient solution on the growth of lamina. We have to use the same method that they have used. Okay, like counting the number of leaves. What, okay, what should be our method? Hmm. 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 Okay, so here we have what is our limiting, like, sorry, independent one is the nutrient concentration, right? Then same volume of nutrient would be okay. Wait, listen to me. Independent is the nutrient concentration, dependent is the number of leaves. Okay, that we count later on. So the constant variables are volume of nutrient solution, starting number of leaves, same temperature, same light intensity, even same carbon dioxide concentration. You can write here like same, sorry, same carbon dioxide concentration. Okay, and then how do we carry out the experiment after certain number of days, whatever days are, or each day we calculate the number of leaves in different nutrients, and then we can plot a graph. Okay. Okay. Hmm. Yes, yes, yes. Or otherwise, you can write would be kept constant. Okay, same number of leaves at the start of the reaction, same volume of nutrients, same temperature, same light intensity, same carbon dioxide concentration. Okay, or otherwise, you can write carbon dioxide concentration, temperature, number of leaves at the start, or the volume of nutrient solution should be kept constant. Okay. Okay, figure 3.1. It's about cells. One cell is plasmolized, other, okay, both, the other cell is not. Okay, state three items of apparatus that you are going to use to view the cells. Okay, this one is quite small. Microscope. Okay. Okay, so microscope cannot work without light, right? Hmm. Slide. 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 Prepared, we can use slide, we can use microscope, and we have to use the light source. For example, lamp, it's a light source. uh, Without light you cannot see, because right now we are using light microscope. Make a large image of only these x and y, or y only. Okay, x and y only. Don't make all the cells, make only these two cells. Okay, x and y. You need to be like very vigilant, or you should have, what shall I say, vigilant eyes. So what are they asking? You have to draw that only. Okay. Describe how would you treat cell from a plant epidermis so that they become plasmolized. Why do cells become plasmolized? When we place them in a um, it's in chapter number 3. Sorry. Good. Or a concentrated solution of salt or sugar. Then, okay, first word, the very key word is the concentrated solution, and the second one, concentrated solution of what? Of salt or sugar. Okay. Okay, so what you need to do,